Right. So again, thank you very much for the opportunity to share a bit about the research that's been going on in my lab for the past 10 years or so. So as mentioned by Sudat, the topic is on substructural similarity searching, and what we're going to do with having more functions as the biological structure data is increasing. And basically, that's having more fun with the increasing biological structure data. Um, I, I know this is a 3D-SIG, but I would like to get everyone up to speed just in case there are someone who's quite new to 3D structures. So just as a very quick introduction, the functions of biological macromolecules, proteins, RNA, DNA are determined by the 3D structures that they fall into. And this is sampling from the PDB of some of the structures that are available currently. So very quickly again, the structure of the macromolecule determines its function, and it shows the molecule as it really is. And this allows us to see the molecular interactions that are happening. And changes in the structure can result in changes to the function. Uh, but again, just to get everyone up to speed, what does the structure data look like, and where and how is the data kept? So the protein data bank is basically the repository, the central repository of structure coordinate data for biological macromolecules, basically DNA, RNA, and proteins. And what the data is like is basically Cartesian coordinates in X, Y, and Z that will tell you where the atoms are in 3D space. Okay. When the PDB started in 1971, for the first 13 years or so, there were only about less than 200 entries. But as time went by, during the second decade, it reached about 1,500. And then in the 20,000s, during around the time I did my PhD in 2003, 2004. And now, 10 years after that, in 2013, it's almost 100,000. But recently, about last week, the PDB finally crossed the threshold of 200,000 coordinate structures. And more recently, this week, we have reached about 2,000 structures. Okay. So where is all this data coming from? The majority of it is coming from the X-ray diffraction of protein crystals. Then there are also some from cryo-electron microscopy and NMR spectroscopy. But more recently, there are about 1 million structures of computed structure models that are also in the PDB. And this is a more recent addition. However, as we all know, the vast majority of the data in biology is generated from the sequencing of DNA. There are billions of them available. And we have protein sequences from this because there are rules that allow us to translate from the central dogma. There are rules that allow us to translate that DNA sequence into the protein sequence. So because of the sequence abundance, the most common analysis that we do in bioinformatics is sequence alignment. And means that there are lots and lots of sequences to compare. And we are therefore using sequences to understand the function and evolution of biological macromolecules, which is fine because there are rules that allow for such extrapolations and information transfers. However, we need to consider the fact that proteins and nucleic acids do not look like this. They look more like that image that I showed you earlier. So the sequences do not provide the structural context or any direct insights into the function of the protein or of the macromolecule that the structure allows. Okay. So can we use the abundant sequences to increase the number of structures? So what we know is that the information needed for a protein to fall into the functional structure is already available in the sequence. So can we then compute and predict the structure from the sequence? And until recently, or until 2020, the accuracy of such predicted structures has been very low and quite inconsistent. However, methods that utilize deep learning has proven to be quite successful, and two notable ones are Rosetta Fold and AlphaFold. There more recently, there are quite a number that has gone into this mix, which I'm not going to discuss further here. But just to show how sort of good AlphaFold and these new prediction methods are, I've got an example here of a protein that we solved. This is a hypothetical protein that we solve in experimentally using X-ray crystallography, and this structure was not yet available in the PDB, and there was no known function assigned to it. So in gold is the structure that we solve by crystallography, and in blue is the one that was predicted by AlphaFold. So as you can see there quite clearly that there is very little difference, and most of it superpose quite well, although you can actually see some variations in the loop regions there. Okay, but in general, the prediction was quite good, but not always, especially for intrinsically disordered proteins. In this particular case, I took an example of the D-sub protein, or the damage suppressor protein, from tardigrade or water bears, and it was predicted using both Rosetta Fold and AlphaFold. So in gold, Rosetta Fold; in blue, AlphaFold. And as you can see here, even when superposition was done, it couldn't quite find any similarities. It couldn't really agree on what the fold of the structure is. So it's clear that experimental methods are not going to be replaced yet. About 2 years ago, in July of 2021, AlphaFold computed about almost 400,000 structures from 21 organisms that are commonly used as models in biology. And of that number, about 20% of it have or carry the label as having uncharacterized function. And more recently, in 2022, AlphaFold predicted or computed the structures of 200 million records that are available in the UniProt database, and of that, roughly about 54 million, or 23% of it, are labeled as having uncharacterized function. Now, in the PDB, after 52 years, we have 200,000 structures, and of which about 17,000, or around 9%, have unknown function. In addition to proteins with structures that have unknown functions, a large number of proteins may have known functions but have never had the structure solved. So these are proteins that have known function but may have an unknown mechanism. An example of these are perhaps proteins that we know to cleave DNA, for example, from assays, but we do not know how it is cleaving the DNA. Okay. So using computational structure modeling, in addition to the experimental methods, we now have more structures to analyze and understand. And as a result of this, we are spending less time and effort on the prediction process and more time on where it matters, which is on the analysis and understanding of the functions of these structures. So we're now getting a flood of structure data. So how do we go about assigning functions to this flood of structure data? Obviously, we can experiment and assay them, yes. But where do we start? And what can be done to analyze protein structures to determine their function? So in general, we can explore the surface, we can look at the physical chemical properties, we can of course compare structures, and there are many methods that are available for this. You've got DALI, you've got FOLSEQ, okay? But what I'm going to try and do today is we're going to look at how a subset of these residues in a structure can be used to perhaps assign functions. So the overall structure of proteins and nucleic acids are important for their functions, but only a subset of these residues are going to be directly involved in a particular mechanism. And typically these functional residues can be conserved as 3D substructures, and they are conserved even when the rest of the fold may be different. Okay. So if we can find 3D substructures with known mechanisms in structures with unknown functions, the mechanisms that the structures are involved in could be similar. So we could then transfer what is known to the unknown. Okay. A good example of this, I'm going to use a very simple example, is the serine protease catalytic triad that we can find in two unrelated proteins, chymotrypsin and satylysin. In this particular case here on the left of the screen, you can see the catalytic triad in chymotrypsin, and on the right, you can see the ones from satylysin. And clearly the arrangement of the residues involved in this catalytic triad is similar, where you've got the aspartic residue, histidine and serine, and the same arrangement be happening in satylysin. And yet, there is no full similarity between these two proteins. So it's clear that different enzymes can carry out catalysis using the same reaction mechanism, even though they are not related. So just to sort of define what I'm referring to when I say 3D substructure, in this work, or in the work that I will be telling about, it basically refers to constellations of residues that are components of a complete or a larger macromolecule. So, for example, for a protein structure, it's basically a cluster of amino acid residues. For RNA, it could be a cluster of base residues. And if there are repeats of this, then they will also be known as 3D motifs or structural motifs. And in order for us to be able to do the searching and comparisons of these substructures, we use graph theory. And very quickly, for those of you who are not familiar with graph theory, a graph is said to consist of nodes and edges. And the edges describe the relationships between the nodes. And let's go back to that serine protease catalytic triad. Try to look at it from a graph theory point of view. So in this particular case, the side chains of the amino acids can actually be the nodes of a graph, and the distances between them define the edges or the relationships in terms of spatial distance. Okay. So we can represent the side chains of these amino acids as graphs, where what we call them, where we refer to them as pseudo atom, because we're taking a graph and representing the whole side chain as a pseudo atom or a pseudo atom vector. This is just a single representation, which is a simplification of the amino acid side chain. So going back to this, so we can represent the side chains of these amino acids as a graph. We can then also calculate or measure the distances between them, and from there we will be able to get values as to what these distances are and build a matrix from those distance values. And this is the matrix that we will use to solve the graph theoretical algorithm. So in many of the cases that I'm going to be talking about, these algorithms use an existing algorithm, and it solves the subgraph isomorphism problem. Although in some cases we solve maximum click searching problem. And there are actually very fast subgraph isomorphism searching algorithms available. So those are not something we developed. Many of these are something that have already been in existence since the 70s. One example of subgraph isomorphism that we use is the Ullman subgraph isomorphism algorithm. And another one that we use is the Brown and Kabosh maximum click searching algorithm. Okay. So that substructure, for example, the serine protease catalytic triad just now can be used as an input to find occurrences of the same or similar arrangements in other protein structures. So we can take that the 3D information about the amino acid arrangement, convert it into the matrices. But of course, we need to adjust for the fact that the spatial arrangements are not going to be exact matches. So we add in a distance tolerance, and a common distance tolerance that we use in our searches is 1.5 angstroms. However, sometimes we also use a percentage. I'll show you later. For RNA, we'll be using a 30% distance tolerance percentage. Okay. So once we have the query, we also need to convert all the other structures that we want to search against to become the database into the same graph scheme. And what we can then do is imagine if this is a database, we would then be able to find occurrences of that particular catalytic triad in the database. Okay. So you might think that surely that is an obvious problem and a rather obvious solution. And the answer to that is yes. But if we go through the literature, what you'll find is that most of the other methods will use the C-alpha backbone, whereas our method uses the representations of the side chains of the amino acids. This has advantages because quite a lot of the functions being carried out by these proteins will be carried out by these amino acid side chains and not at the point of the C-alpha. And to demonstrate that, we can show you here an example where you can see that the side chains superpose, although the C-alphas are quite distant. So if this were a functional arrangement, then the function will be preserved or conserved, although as you can see here, if you were to carry out a C-alpha search, it will not be able to retrieve this particular match. Okay. So those algorithms that we develop, we made them into programs that are web accessible, and one is called Sprite, which takes a whole structure as an input. It can be in the format of a PDB file or an MMSIF file, and then it searches a motif database to find similarities. In reverse, we've got another program which is also web accessible called ASAM, which takes the input of a 3D arrangement in PDB format, and it then searches a structural database or PDB datasets. So we can find the 3D substructures in a database of protein structures. We have done the test and we are able to do that. So what can we do with that capability? So one of the things that we tried looking at is whether we can take known drug binding sites and then find similar binding sites in other structures. And those other structures might be might be proteins that are not known to be a target for that particular compound. So in effect, what this allows us to do is it allows us to carry out drug repositioning, where we're able to try and see or explore whether an existing compound can bind to an alternative site that is not, or that may come from a protein that has a totally different fold and may be unrelated to the original target for that drug compound. And this is an example of what we did for the COVID-19 structures that are available in the PDB. One thing of interest is that since we are able to find all these off-target binding sites or alternative binding sites, that also means that if a drug binds to an unintended target, it may cause other effects, especially if it's in the host. And these could include side effects that we know to occur, including some level of toxicity. So we could use the same method to explore whether a drug that is repositioned could result in any potential side effects. And at the same time, we could also use this method to explore the human proteome, whether a drug that is in development, where we have the binding site data, we can then search the human proteome and decide whether there are potential off-target binding sites that could cause toxicity. So this could form a sort of a preliminary screening that allows for drug discovery to be sort of screened beforehand down to clinical trials. So having said that, knowing the off-target sites is not a bad thing. So what we're trying to do is we're trying to make use of this, and we're trying to go through the available list of side effects that we can find in drug-associated databases, and see whether we can actually assign the side effect that is observed through clinical trials to something mechanistic. For example, if it's known to bind to a particular protein that can cause something, previously it was just an observation and the mechanism of how that was happening is not known. We hope that we could provide some insight as to how that side effect is occurring. And in a way, we're hoping that in the future, this sort of perhaps provides the necessary intelligence to clinicians and trials programs to avoid surprises during clinical trials. So we've got known drug binding sites, and we try to find similar binding sites in other structures. But since we're able to find, or we're able to know what that compound is, perhaps we can also see whether we can stick an alternative compound that can bind to that similar binding site. And we're hoping that there is available literature and evidence that, in a way, different compounds can increase the efficacy or increase tolerance among different individuals. So this allows for targeting of the same site to continue with structurally similar compounds should the original compound be deemed unsuitable, or in some cases have low tolerance for some patients. And this is an example of the work that we had done, where, as you can see on the right panel there, once we identified what the alternative target site is, we then look at the compound that can bind to the alternative site, and then we try and find other similar compounds to it. This also opens up avenues to allow for new compounds to be designed that could better fit the alternative target site. So what else can we do with this capacity? And one of the things that we have been or that other people have been using our substructure searching service for is to identify similar mechanisms in diverse proteins, especially in cases where there are no detectable sequence similarity and there are no detectable fold similarity. And we find that this is something that is being used a lot by structural biologists and structural genomics groups. And in this particular case here, it's led to the discovery of a novel motif of a four-aspectic arrangement that was first found in acetylcholinesterase from a stingray, if I'm not mistaken. And it was found to occur in other enzymes as well. And this was published in 2021 by an unrelated research at that time to us. And we then started collaborating on that particular work up now. Another thing that sort of surprised us is that this particular tool that we developed for substructure searching is actually in use by several universities in Europe and the States as part of the basal biochemistry curriculum. And this was quite a surprise because we never actually designed it as a teaching tool. And but somehow it became a teaching tool to teach biochemistry, especially on catalytic site analysis. So we can find this in what I've been showing you are searches and discoveries that are happening for experimental structures. So how useful is this for computed models, especially in light of the flood of AlphaFold structures that are now available? So here I'm showing basically the same protein, which is called Burkholderia lethal factor 1 or BLF1. And on the leftmost panel there is the experimental structure that was solved to a very high resolution, 1 Armstrong resolution. And as you can see there, using our program Sprite, it managed to match the arrangement or the motif that can be found in BLF1 itself. So 3TU8 there on the left panel that you can see is actually the original BLF1 structure that was solved in 2011. And what you can see here, 6RVU, is a more recent structure that was solved as part of a complex. And as you can see there, second on that list is also is a protein called chemotaxis methylation protein. And what this is actually both BLF1 and the chemotaxis regulator are actually deamidase enzymes. Okay. So in the case of BLF1, it deamidates a glin to become a glue, and that deamidation process results in loss of protein translation in the host. Sorry, I forgot to tell you that Burkholderia is actually a pathogen. So this particular lethal factor is actually a toxin, where it deamidates the human EIF4A translation initiation factor, resulting in the glin to become a glue. Okay. And in the middle panel here, what you can see here labeled there is BPSL 1549 from the AFDB database. And if you're familiar with how AlphaFold sort of calculates the reliability or confidence of these structures, is that the darker the blue shades, the higher the confidence is, and as it goes to yellow and amber, the lower the confidence of the PLDDT values is. So using the sequence of BLF1, the AlphaFold pipeline that is being used by EBI actually came up with this structure. And in generally, it has a low confidence. And as you can see from the sprite search, it's quite different from the actual experimental structure. So it's not turning up the right the right residues for the active site. And this is not unexpected because AlphaFold itself thinks that it got the structure more or less not right. So here we can see the value that AlphaFold actually sort of knows quite well when it is going wrong. But I don't quite have an explanation as to why it's not getting a structure that is actually available in the PDB quite accurate. And all I can, at the moment, what I can think of is that some pipeline that generalizes how the structures are being computed is actually messing up some of these, even though there are actually examples available in the PDB. But that doesn't mean that AlphaFold is getting it wrong. So as I was saying, I think it's the pipeline that's getting it wrong, because as you can see on the rightmost panel, we took a very remote homologue of BLF1, which is we call XLF1, which is a Xanthomonas, a little factor 1. We do not know whether it is a toxin or not, but we named it as Xanthomonas little factor 1 simply because it is a BLF1 homologue. Although it's only got 19% sequence identity to BLF1, when the structure was computed using AlphaFold, you get more or less a very similar structure to what you can see for our 6RVU on the left. And as you can see from the sprite searches, it retrieved more or less the same results as the actual experimental structure. And as you can see from the superposition there, the query being the one in CPK coloring and the match being the one in green, it is superposed quite well for the active site of the active site of BLF1. Okay, so clearly that here although these are computed structure models, we are able to sort of use these programs to actually annotate computed structure models, provided that they are of sort of a high confidence or with high PLDDT values. But that's assuming that we know what it is that we're supposed to match. So we have an idea of what it is that we're looking for, or we have a database of knowns to match against. What if we don't have an exact idea of the arrangement that we are interested in? And for that, we came up with a more conceptual approach towards searching for these substructural similarities. In this particular case, instead of providing an actual pattern or an actual 3D arrangement as the query or as the input, what the program takes in as input is actually a hypothetical 3D arrangement, where you can define what residues you are interested in and the relationship between them being the distance. Okay. And you can then search whether there are other PDB structures or whether there are other protein structures that contain such an arrangement. This is actually much looser than having a rigid query where you know exactly what it is that you're searching for. And interestingly, we were able to find something that even when we quite loosely defined it. And in this particular example, you can see on the left in the panel A, one protein and in cyan and another one in gold, and in the panel marked as B, you can see that this forms a tetrad where it's got a histidine, and a cysteine arrangement that acts as a metal or a zinc binding motif. But clearly from the left-hand panel there, you can see that they are totally unrelated uh, in terms of evolution, despite being able to form that same zinc binding site. And if we were to look at the sequence as well, the sequence alignment is totally off, and we can't actually see any particular arrangement um, that shows that these are homologous proteins. Interestingly, using Imagine, we were also able to find novel motifs that are present only in a 3D context, where these particular motifs are actually single residues uh, at the sequence level. But when looking at an assembly, you'll find that these are these assemble into a motif. So the example here that we can see here is a histidine in the middle there, histidine. I think it's a 6 histidine arrangement that I did a search for just on a whim, where I just decided that can I find an arrangement of 6 histidines that are roughly about 4.5 angstroms apart from each other, and are there any examples in the database that contains such an arrangement? And as you can see here, there are actually assemblies that contain this particular arrangement. And if we were to take it apart, you'll find that each structure might contribute this 2 histidines, and the one on top there, the glue is being contributed by only one glue from each uh, monomeric subunit. And as a result of that, if you were to stretch it out into the sequence, that motif is going to be lost because it is only present in a 3D context. So the interesting thing would be, can we transfer this knowledge to sequence level searches using machine learning, and from there be able to also improve algorithms that deal with assembly of these proteins. So just to sort of recap and summarize, we can use the 3D substructure similarity searching to find similar substructures in unrelated or highly divergent proteins. And we have shown that we could use it to explore the potential for drug repositioning or off-target binding. And we are also able to find novel arrangements based on similar chemistry. So that's for proteins. What about other biological macromolecules? Can we use a similar approach for RNA structures? And if we were to look at the RNA structures that are available in the PDB, this has lagged behind quite a lot compared to proteins, where there are 100 over 1,000, 200,000 structures already, and most of them are proteins. There are only about almost 7,000 entries with RNA chains in the PDB. Although if you were to look at this chart, the pace of the availability of these RNA structures started to pick up after the 2000s, when the first high-resolution structures of the ribosomes became available. But even now, there is a lack of tools that are able to analyze the structures um, for RNA, and perhaps one reason why that is so is because there are not a lot of structures to deal with, and perhaps at the moment it's not of much interest, although we're starting to see interest for RNA structure analysis picking up. One thing worth noting is that RNA structure prediction is not quite the level of protein structure prediction that we can see accuracy like AlphaFold and Rosetta Fold. So using a similar scheme like we were using for amino acid side chains, we're able to actually use it also for RNA bases. And in this particular case that you can see a very similar arrangement. So instead of a catalytic triad, I've got here a base triple, and we represent so the bases as pseudoatom vectors. And again, we use the distances between those nodes, which are the pseudoatom vectors, as the edges of the graph, and then we convert it into matrices that we can then start searching or comparing using graph theory. In this particular case, the input is an RNA chain containing structure. It can be either in PDB format or the ribosomal assemblies are usually in MMSIF. And it then searches a structural data set or a PDB data set. Okay. And we have shown that yes, we are indeed able to find even novel motifs using that particular search scheme, which is basically what we transferred from our capacity to search for protein structures using amino acid side chains. So we went through the whole gamut of it. So we can find similar arrangements. Can we then use this method to identify drug repositioning sites for drugs that target RNA molecules? And in a way, yes, we can do it, but it is not quite as clear cut as it might be for proteins. So that's still sort of work in progress that we need to look into. But one thing that I would like to sort of before I end this is to share a bit on this particular query that we start using a known base triple, which is basically a UAU base triple that I think was already reported from as early on from the work of Ignatius Tinoco Jr. In it could have been in the 80s and 90s. So this particular base triple is known. It's not a novel base triple by itself, but what we were able to find is a novel context for it. So as you can see on the right there, this is domain 5 of the large subunit of the ribosomal RNA assembly. And what we are able to find is a UAU stacked on top of another UAU. And as you can see there, the adenine in the middle there is actually opposite directions. Okay, but it is a UAU which is stacked one on top of the other, and you can actually see here highlighted in red where it is in domain 5 of the LSU of the large subunit of the ribosomal RNA assembly. And you can see here 3 arms, one extending up here that you can, if you can follow my laser pointer, one is going up, one is going down to the right here, and one is going off to the left here. So that sort of arrangement, or that UAU, that stack UAU motif, seems to be holding together these 3 arms of this domain. And this is another view of it where you can actually see the stacking better. So you've got UAU there and a UAU there. And again, this is where it is. And when we first found it, seemed interesting. So we decided to see how conserved this particular arrangement is. And as it turns out, all available all available ribosomal RNA structures that are complete and have this particular region will actually have this particular UAU stack. It doesn't matter from what organism it's from, whether it's human, whether it's archaea, or whether it's yeast. So and it also includes the mitoribosome. So all these diverse RNA structures contain this particular motif. So could it be could it possibly be a universal motif that's hidden in plain sight? And in this particular rendering, I'm just showing again that particular UAU stack, which is here on top of this arrangement, and it's holding the three arms, as you can see there, if you were to follow my laser pointer. And there, what we can see is the stack UAU, and here this cluster of bases is the peptidyl transferase center for the ribosomal RNA. And ignore this for now, because it's basically our own notes trying to figure out what's happening in these structures. So we decided to see how diverse this is by extending it to sequence level as well. And we were able to show that all sequences from that are available in all these different kingdoms of life, and in all these diverse organisms. So as long as it's got a complete ribosomal RNA, we are sort of able to find that particular UAU arrangement in the sequence. And for those with structures, we're able to find it in the structure as well. So just to take it off a bit, what if by using this, or by using that particular motif, we're able to come up with a minimal ribosome, and this is the concept of the proto-ribosome. And in this particular image, in this particular figure here, this is taken from the work of Ada Yonaf and co-workers regarding the proto-ribosome concept, published almost to a year, almost to the day last year. And we decided to take that proto-ribosome model that they have proposed and see where this fitted, or this UAU stack fitted onto that proto-ribosome model. And here you can see the UAU stack on the left-hand corner here. But what you can see there is that only 3 of them are actually in the model, which is the ones labeled where the residue numbers are labeled in green, but the other 3 are not in the proto-ribosome model. And if we were to add in the peptidyl transferase center there, you also see that 2 of these bases that are known to be as part of the PTC are not quite in the proto-ribosome model. So is it possible that we might be able to improve the model of the proto-ribosome by including this sort of a very universal motif that can be found in all available ribosomal RNA structures to date? So that sort of ends what I wanted to share with everyone. Just a bit of shameless advertising here. So we've got, as I've gone through just now, we've got web servers of protein substructure similarity searching. So we've got 3 programs there: Sprite, ASM, and Imagine. And here, just to recap, one takes a whole structure as input, and in either PDB or MMSIF, and it searches a motif database. While the ASM does it in reverse, it takes the motif and searches a structural database. Imagine takes a hypothetical arrangement and then searches a structural database similar to ASM. And then we have a web program that sort of integrates both Sprite and ASM, and which is specific for exploring drug repositioning possibilities. I mentioned we also develop tools for RNA-based substructure similarity searching. I mentioned NASAM. which takes a whole structure in either PDB or MMC format, and this is what we use to actually identify that potential universal UAU stack motif, and it searches a motif database. But we have also got two other tools. One is Cognac, depending on how you want to pronounce it, um, which basically searches or takes hydrogen bonding connectivity data, and it searches an RNA structure database, which I did not cover. And we've got a database that takes either a no motif query or text, and it searches an RNA structure database. And this one also I did not cover in this seminar. We've had problems with users over the years, not sure of which particular tool to use, or using the wrong tool for their searches. So we came up with one site to interface them all, which we call graphs, graph theoretical applications for substructure searching, which sort of guides you through the process of which particular program to use without you actually needing to know what program it is that you're using. So it basically asks you what's your query molecular structure. And if you answer protein, then it takes you through the rest of the steps until you get to the point where you want to end up. So that's the end of my shameless advertising. Just to conclude, the 3D substructure searches in biological macromolecules can be carried out by representing the amino acid side chains or bases as graphs, and we have demonstrated that to be quite useful. And such searches can allow for 3D substructures to be found in a database of structures or vice versa. And the increasing availability of protein and RNA structures will be the next frontier, in my opinion, will be the next frontier towards understanding the extent of atomic level mechanistic diversity or conservation. And hopefully using these tools, we will be able to assign more functions and discover new biology. And of course, having more functions means having more fun in front of the computer. All that work I could not have done without this very talented group of individuals. Many of them have gone on to better things. And of course, I've over the years collaborated with many of these people. I think I mentioned the work with Professor Sussman and Professor Israel Silman at Wiseman just now, who used our programs, and they were actually instrumental in coming up with the idea for graphs, and they provided a lot of feedback that allowed me to come up with our graphs web server. And of course, these funding resources that have funded our research over the years. I think that's the end of my sharing for this morning, evening, and night. So thank you very much. I'll stop sharing now.
Thank you so much, Firdaus. It was very insightful, and you actually went from protein and all the way to RNA. That's a lot of things. For all the audience, I think if you guys have the questions, feel free to drop the questions in the Q&A sessions. So if there's any burning questions, feel free to ask there. Maybe I'll start by asking a few questions. I'm just curious, how fast is your method for the substructural searches?
All right. For searching a database of motifs is quite fast. Takes within 1 or 2 minutes. So if you've got a structure, so it depends basically on how large your structure is. And if you would search a database, if your query is a motif and you want to search a database of the PDB, for example, we use a non-redundant data set. So it's at 30% sequence identity as a starting point. So we don't go very high because you get a lot of redundancy. However, we do get requests from users for a more redundant database. and we can actually generate it for them and put it on the website as well. So I think there are special requests that you might see on our website, specific families of proteins,
Mhm.
specific data sets that, um, if you're not familiar with it, you might, why is that data set there? Because it doesn't make sense for it to be there. But sometimes we've got users who ask for it, and we're more than happy to put it up for them. And the reason why rated like that is because if we were to use the whole PDB, it would slow things down quite a lot. So we have very specific data sets to search against. Um, and it would be lovely if we could actually host all of AlphaFold data, the predicted models, but unfortunately we do not have the resources to do so at the moment. Um, so how fast is it for the SM, or where the query is the motif searching against a structured database? It can take a few minutes. It's longer than the Sprite ones, probably 5 minutes or so. Depending on how large the structure is, the larger, how large the database is. The larger the database, the slower it gets, obviously. Um, but in general, if you're running it and you're finding that it's been running on for 2 3 hours, do drop us an email. Probably something went wrong, and we'll be happy to troubleshoot it for you.
Cool. Thank you so much. And I think there's a is there a downloadable versions of
Um,
software?
yes, sort of. If you have to ask for it. We have given quite a number of groups the downloadable version of it so that they can run their own databases. And we give it as is, without, we don't really need to know what database you're running. um, but we sort of want to know that you have it. So if you want it, there's usually a request for it, and we give it to them, because mainly the installation process can be a bit messy. I don't know why, but it's always been messy. um, it seems to be very particular about specific machine setups. So you might have to do little bits of uh, uh, messing about the parameters to figure out how it's going to work on different machines. So when we know that someone is actually interested in it, we sort of help them through it until it gets running, and then you're on your own. If you don't want to involve us anymore, that's fine. But if you want to work with us on it, that's also fine.
Cool. We have questions from the audience, so you can just read that from the Q&A sessions, right? There are papers that contain binding data for hundreds of unintended off targets of some drugs thought to be specific previously. For example, off target toxicity is a common mechanism for actions of cancer drug undergoing clinical trials. Did you try to validate your predictions against a long list of off targets?
Right. So interestingly, I will not call these predictions because they're more matches. So there isn't really, it's not really predicting anything. It's just matching something. So if you say that this is an off target and it matches to an off target, it probably is the same or a similar thing, but then you have to decide what it is from the biology or from what you know about it. So back to this question is, have we tried searching against that? No, the answer is no. We have wanted to, but we are very short-handed, unfortunately. But have we validate this? Um, we One problem with this is that we're doing the computational upstream end of this, and looking at whether the binding can actually occur, and whether what we are saying could be cytotoxic is actually cytotoxic. So we have recently started working on this, where um, everything that we say is binding is sent off for structural characterization to see that it's actually binding. And anything that we say is binding is then also sent for cytotoxicity studies. So we're trying to develop sort of a drug repositioning pipeline, if I can use that phrase, um, sort of identify, then validate whether those bindings can actually occur, and then see whether they are of potential toxicity. At the moment, just in cell culture, we've not gone beyond that. That is a recently started work, so yes, uh, but yes, an interesting aspect of what we want to do. And previously, we have not really explored it because we have not had the resources to do so. But until recently, it seemed like a good idea to follow through instead of just saying that, hey, this could stick, this could stick, this could stick, but not really following up on any of those hypotheticals. So yeah.
Okay. I think it's quite late now, but I'll just ask one question. I'm kind of curious more about the motif you've mentioned. There's one called 4D. Why is it called a 4D? Is it because of a four-dimensional kind of like structures
Oh
or
yeah,
what?
yeah.
What? Yeah.
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 It's just for for aspartic for
Ah,
aspartic acids.
okay, okay.
So it's binding to a metal, and it was first found in an acetylcholinesterase by I think Professor Israel Silkman and Joel Sussman at Weizmann, and they've got they've got interest into how to move that forward, but I didn't really ask them permission to share about what they're doing, so I'm not going to mention that.